Hello everyone, my name is Bridgetlyn Campaner and I am your reporter for this morning. Have you ever wondered, whenever you bathe in your bathroom or you use the comfort room, you can see some medyo brownish na color, yellow-brown sa toilet bowls or sa bathroom tiles. It is caused by a bacteria called Serratia marcescens. So this is our topic for this moment. Okay. Serratia marcescens is a member of the genus Serratia, which is a part of the family Enterobacteriaceae. Its general characteristics are Serratia marcescens is a gram-negative rod-shaped facultative anaerobic bacterium that is extremely motile. When it is stained, it shows short, pink, colored dot, and it is in a rod-shaped bacteria or bacilliform. As you can see in the picture, bacilliform din siya. So it was discovered by Bartolomeo Biggio in 1819. Serratia marcescens was thought to be harmless at first, but it became a nosocomial opportunistic pathogen. It produces a red pigment, which is described as masqueraded blood, and it is used as a biological marker of infection. So out of 14 species of Serratia, 8 species are associated with human infections. And it is the most common clinical isolation and the most important human pathogen. It can be seen on the tile and shower corners, water lines, and toilet bowls. It appears a pink or orange filmy residue. So those toilets na seldom lang ginagamit is the breeding ground for the Serratia marcescens because of the standing water in the bowl. So Serratia marcescens mag-grow siya in those damp places. So it can also be found in the soil and in human and animal feces. Epidemiology. Serratia marcescens can cause infections in both children and adults. It also ranked 5th among gram-negative organisms in the intensive care unit. It is the 7th most common cause of the pneumonia in Europe and Latin America. Pathogenesis. The Serratia marcescens was associated with low intrinsic pathogenicity. Its isolates produce extracellular products, and it also secretes a pore-forming hemolysin called Shia. Spectrum disease. Exposure to Serratia marcescens may cause urinary and respiratory infections, endocarditis, osteomyelitis, eye infections, wound infections, and meningitis. So, the subclinical manifestations also include cellulitis, abscesses, fluctuant nodules, etc. And it can be transmitted by direct contact. Infected patients who develop urinary tract infections, the symptoms can be itching, burning, discharge, and pain in the kidney. So in children, infections are in the gastrointestinal tract. The clinical conditions in newborn ranges from asymptomatic to meningitis. So the time for diagnosis of Serratia marcescens meningitis is about 0 to 27 days. So the mean time is 2.8 days. And antimicrobial therapy can be given within 48 hours of diagnosis of Serratia marcescens meningitis. So the mortality rate of this bacteria is high, especially if those patients develop endocarditis or meningitis. Significant cause of healthcare associated pulmonary, urinary, and surgical site infections. Also, is a highly resistant to antibiotics. Laboratory diagnosis. So, oxide McConkey is used to diagnose this bacteria. It is a selective and differential culture medium for bacteria. It is designed to selectively isolate gram-negative and enteric bacteria and differentiate them based on lactose fermentation.
In specimen collection, to assure that the laboratory results are accurate, it is important that the specimen be ordered and collected and processed properly. And in the specimen collection and transport of Serratia marcescens, the specimen can be obtained from various samples like blood, urine, and health and environmental cultures. Surveillance cultures such as pharyngeal swabs, nasal swabs, sputum, and stool cultures from all the patients to trace the source or reservoir of the infections. Specimen processing. Body fluids specimen processing must plate as soon as received blood culture bottles and incubate at 37 degrees Celsius. And rectal swab for 38 hours or store at 4 degrees Celsius. And stool culture for 74 hours or 4 degrees Celsius. And sputum bacterial aspirate for 24 hours or store at 4 degrees Celsius. All clinical samples, including the blood culture bottles giving positive signals cultured at the McConaughey agar, blood agar, and chocolate blood agar, plate incubated at 37 degrees Celsius for up to 48 hours. Indirect detection methods. All Enterobacteriaceae, including the Serratia marcescens, have similar microscopic morphology. Isolation of gram-negative organisms from a sterile site, including the cerebrospinal fluid, blood, and other body fluids may assist the physician in prescribing the accompanying therapy. In cultivation, in media choice, bacteria grow well on routine laboratory media such as 5% sheep blood and chocolate and McConaughey agars. In addition to this media are the selective agars, Hectoen enteric agar and psilocybin dioxide chocolate agar are commonly used to cultivate enteric pathogen from gastrointestinal specimen. It is mentioned here about the use of the gastrointestinal specimen because the gastrointestinal tract is recognized as the predominant site of colonization for the Serratia marcescens, as is the case for most members of the Enterobacteriaceae family.
So, approach to identification. Serratia marcescens is a species of rod-shaped, gram-negative bacteria in the family of Yersinia. It is a facultative anaerobe and an opportunistic pathogen. It is commonly involved in hospital-acquired infections, particularly catheter-associated bacteremia, urinary tract infection, and wound infection. So, the serodiagnosis of Serratia marcescens is BBL intertubesecond, API 20E strips, blood agar, McConaughey agar, and specific real-time PCR assay. S. marcescens are susceptible to ampicillin, ceftazidime, cefotaxime, ceprofloxacin, tazobactam, gentamicin, ertapenem, imipenem, colistin, and amikacin. S. marcescens is known to be transmitted hand-to-hand contact. So, enable for us to prevent this bacteria, we have to disinfect environmental hand contact surfaces and multi-use equipment, enforce workers to wash and disinfect their hands before and after patient care, use new gloves for each treatment, use chlorine bleach spray, periodically investigate the environment, implementing strict hygiene policies, and promoting proper hand washing. So, fact. Patients who develop an infection have legal rights to seek financial recovery for their damages, bruises, and injury. Hospitals should develop and enforce effective policies to minimize dangerous bacterium by sanitizing surfaces, promoting hand hygiene, and enforcing equipment or fixture disinfecting policies to minimize every patient's risk of being injured by contaminated surfaces. So that's it for our reporting, and thank you for listening.